We talked about it in the anaerobic aspect, where we have a bacteria where its desire is to get or make some kind of ATP. And we talked about this in the process of fermentation. But we also mentioned that in the process of making these 2 ATP from fermentation, we're also going to get 2 NADH. And these 2 NADH will help cycle back to the bacteria to repeat the fermentation process. And what we didn't talk about is bacteria that have the ability to use air to generate more than 2 ATP and 2 NADH. Now, whether the bacteria is an anaerobe or aerobe, it will always start off with this process. But if it is able to use air and has the presence of air, it will go down a cascade of events that will allow it to generate more ATP, specifically 32 ATP. So, how does it get here? Well, from here, we already know that our bacteria started out with a glucose molecule.
Now, guys, when you're watching this, he's going to tell you 32 ATP. We're going to go with the number 38. That's the maximum amount of ATP that can be produced. So when I go over stuff in your notes, don't get confused. Different bacteria make different amounts, and it varies from 28 to 38. And he's just kind of taking an average here of 32. So that's why that number is going to be different. When we discuss this in a minute, it will be 38. All right. I want to make that clear to you.
Which is a 6-carbon molecule. It will then break it down through a process called glycolysis. As we've already mentioned before, with anaerobic respiration, we start off with glucose molecule. This glucose molecule will go through a process called glycolysis. And once we're done with glycolysis, our glucose will be processed into 2 pyruvate molecules. But in that process of glycolysis, we get our 2 ATP and 2 NADH. Now, what happens after this step can only be achieved by bacteria that are aerobes. Although the next step that we're going to go over, pyruvate decarboxylation, is technically still an anaerobic step. We take these 2 pyruvate and put it through pyruvate decarboxylation, which will convert our 2 pyruvate molecules to 2 acetyl-CoAs. And just like glycolysis, pyruvate decarboxylation gains us some energy here in the form of 2 more NADH. Now lastly, we're going to take our 2 acetyl-CoA molecules and put it through the last process of the electron transport chain, Krebs, or TCA, all of them meaning the same thing. And once our 2 acetyl-CoAs have fully gone through the electron transport chain, we can expect to get 2 more ATP, 2 FADH2, and 6 NADH. And now once we add all these things up together, you can see that we have 4 ATP, 2 FADH2, and 10 NADH. But you might be saying to yourself, Fritz, I thought we were going to get 32 ATP, not 4 in these other molecules that you mentioned, and I'm not sure about what the significance is. Well, their significance comes into play because we actually don't put the 2 acetyl-CoA through the electron transport chain. They do act as reactants, but what we're also doing in the electron transport chain is transporting the electrons from FADH2 and NADH. And once those electrons have been transported, we can expect that 1.5 ATP approximately and 2.5 ATP will be generated for each molecule. So this comes out to 3 ATP, and this comes out to 25 ATP. So no need to whip out your calculators, it's just simple addition. 4 plus 5, 29 plus 3 is 32. And that's how we get 32 ATP. Now, depending on the textbook and the actual processes that the bacteria go through, this number can vary, but you can expect it to be anywhere in the realm of 28 to 36. Because this is a microbiology class and not a biochemistry class, I really think it would be extremely cruel for your teachers to give you something like, how many ATP is created at the end of the electron transport chain? Is it 28, 30, 32, or 36? That to me would be extremely rare. In my experience, they'll give you some numbers that might have some correlation or no correlation at all, but they're going to be very off from this range right here. So
I'm going to go ahead and stop that here. He's just trying to tell you what test questions should look like. Again, with us, let me get my camera back on. All right, so with us, again, we're going to be 38. Now, you're going to see in the notes, I'm going to go over basically what he just went over with you. I just kind of wanted to let him kind of go into it a little bit so you kind of understand what's going on. And then when I talk about it, you can kind of come back and relate to this. So we're going to talk about glycolysis, and we're going to talk about the Krebs cycle, and we'll talk about the electron transport chain, right? Those are the 3 steps in cellular respiration. We're also going to talk about fermentation later. He kind of talked about it first and then went into the aerobic. I like to show you the aerobic first because that's usually the preferred method of making ATP, because you can get so many ATP from 1 molecule of glucose. When you go through fermentation, you only get 2. So during aerobic respiration, we're going to get that 38. So that's more advantageous for the organism to get the most bang for their buck. When they go through fermentation, they're creating things like ethanol and lactic acid that are high energy molecules, and they're not able to get that energy out of it. So it's kind of a waste, but they only do that usually when they have to. All right. Same thing with our muscles. We don't want to go into lactic acid fermentation unless we're starving for oxygen, and we have to. And then we have to make that ATP that way. So this is just the preferred method, whereas fermentation is kind of secondary. Now, some organisms are strict anaerobes, and they can't go through aerobic respiration. And they have some different pathways that we'll look at. But typically, most of the bacteria that we study are aerobic or at least facultative anaerobes, where they can switch back and forth. All right. So let's move on from here, and I will kind of go over everything with you. Now, this is a great chart. Now, in your book, you're not going to see these numbers, but I put these in here just so you would know what's going on as far as the number of ATP. So if you have a diagram like this available, go ahead and write those numbers in. Again, you'll be able to use this on your test. And, you know, the goal of this is not to make you memorize all these pathways, but to understand them and how they work and what goes on with each one. So we're going to start out with glycolysis, which is up here. And that's where we take glucose and convert it into pyruvic acid. Now, we make 2 ATP this way. So we're getting a little bit of energy, all right? So if I'm a bacteria, I'm hopping along, I get 2 little ATP, that might last me a little while, all right? But I need to sustain that, all right? So again, typically I would want to run down this pathway, all right, where I would go through aerobic respiration, go through the Krebs cycle, produce 2 more ATP, and then go down this electron transport chain and produce that massive amount of 34. So again, typically this is the preferred method. So if a bacteria can live in the presence of oxygen, it's going to want to go through cellular respiration. You just get more energy for the amount of food you're consuming, all right? But if we are in an anaerobic environment, what we're going to do here is switch over to fermentation, and we will go into fermentation, and typically we'll produce either lactic acid or ethanol. There are other byproducts of fermentation, but those are the 2 big ones that we're going to look at. Now, I'll give you a chart in a minute that shows you some other materials that can be made through fermentation, but those are the 2 major ones, all right? Now, notice what we're doing here, though, with fermentation. We are going through this pathway for one reason only, and that is to get an electron in the form of NADH to go back into the system again and keep this going. All right. So again, we're going to want to produce NADH, and we've already made the ATP. We're just going through fermentation so that we can keep this cycle going, so we can keep making 2 ATP from every molecule of glucose. All right. So again, this is just a fail-safe. Hey, let's make some energy. Let's get what we can out of this. Times are bad. There's no oxygen around. And let's do this and see if we can, survive long enough until oxygen returns. All right. And that's kind of the way you need to look at it. So this is a great chart because it shows you both the aerobic pathway and the anaerobic pathway in one place. Right. And again, a lot of organisms can switch back and forth. Now, there are some that are strict aerobes, which if they don't have oxygen, they die, like us. If we don't have oxygen, we're strict aerobes. We have to have oxygen in order to survive. But some bacteria have the ability to flip back and forth, right? So again, this is the preferred method. This is kind of secondary in most cases. All right, so let's start out with glycolysis. This is the first step, either in fermentation or aerobic respiration. So this step's going to be the same for both. And what we're going to do is take this glucose molecule and basically split it in half. All right. Now, it takes energy to break those bonds and split that in half. So we have to consume 2 ATP initially to get this whole process started. All right. So we're going to put in 2 ATP and they'll convert to ADP. And we'll use that energy to split this molecule. All right. And when we do, we're going to go through and then produce 4 ATP. So since we put 2 in and we get 4 out, we get a net of 2. That's a gain of 2, all right? And now we have these pyruvate molecules. They can either go into fermentation or into aerobic respiration, all right? So again, we have 2 ATP in, 4 out. That gives us a net of 2. So again, all we're doing is splitting glucose into these pyruvates, and then these are going to go through either the Krebs cycle or fermentation, right? So there's a preparatory stage, again, where we put in those 2 ATP, and then we split glucose, and we basically form glyceraldehyde 3-phosphate. And then in the secondary energy conserving stage, that's where we produce the 4 ATP and the 2 NADH. Now, these are going to be important, all right? These NADH molecules carry energy in the form of an electron. So when we produce these, they're not waste products, all right? These are electron carriers that could carry this electron into the electron transport chain later on. So don't think of these as waste products. Now, I'll mention when things are waste products, like CO2 is a waste product, all right? But we'll look at this total reaction here in just a second. You'll get a better idea of what I'm talking about, all right? So again, we call this the energy conserving state because we are getting a net gain of 2 ATP. So again, the preparatory, we use 2. In the energy conserving stage, we produce 4, and that is going to give us a net of 2 ATP and a net of 2 NADH, which do carry energy for us. All right, keep that in mind. All right, now this is a big long pathway. Here's our preparatory stage. Notice we put in 1 ATP, 2 ATP. We split that glucose molecule, and then that's going to go into the energy conserving stage. And this is where we make ATP. And notice we make 2 here and 2 down here, and we make 2 NADH here that's going to carry electrons. All right. Now we do give off water here. And so when we breathe out, some of that water vapor that we're breathing out is produced in this reaction, all right, along with CO2 later on, all right? So notice here, we're basically just trying to get a little bit of ATP out of this and get ready for the next step, all right? So this is glycolysis. Again, preparatory stage, we put 2 in. Energy conserving stage, we get 4 out for a net gain of 2. So at this point, we have 2 ATP. Now, again, if we are in the presence of oxygen, we can make more ATP. If we're not, if we're in an anaerobic environment, then we're kind of stuck, and we just need to get this process going again. So what we'll do is go through fermentation to make some of these electron carriers that can come back and get the system going again, all right? And again, it's a tough system to run through, but you got to do what you got to do when there's no oxygen available. All right, so again, this is glycolysis. This is kind of a long version of it. So glucose plus 2 ATP plus 2 ADP plus 2 phosphate plus 2 NAD plus gives us 2 pyruvic acids, 4 ATP, 2 NADH, and 2 hydrogen ion. But again, this is the energy investment or the preparatory stage we put into, and then we get 4 out down here for a net gain of 2. And then we have these pyruvate molecules that still contain a lot of energy that can then go either into fermentation if we're in an anaerobic environment or aerobic respiration if we're in an aerobic environment. Now, there's additional pathways to glycolysis. Some bacteria utilize these. We're not going to go into this. It's really kind of the same process, or really we're trying to get to the same point, and that's to get to glycolysis. So again, these just use other ways of doing things to get energy. And again, these are specialized bacteria that go down here. So we're not really going to get involved with that. Just realize there are other pathways out there. Right? I just want to mainly concentrate on the 2 that we're starting to look at, the fermentation and the aerobic respiration. All right, so oxidation of molecule liberates electrons. Remember those redox reactions we talked about the other day. So we have what's called an electron acceptor, right? Remember, when we receive an electron, right, we are being reduced, right? So when we gain an electron as an electron receptor or acceptor, then we're being reduced, and the one that gives away the electron is being oxidized, right? So again, just kind of get this in mind. And when we talk about the electron transport chain, hopefully some of this will make sense to you. I know right now you're probably like, what in the world is he talking about? It'll make sense when I put it all together and kind of show you these pathways. And again, you don't need to memorize all of these intermediates. I just want you to understand what's going on as far as the electrons moving from molecule to molecule and how we're making ATP along the way. Right? That's the main thing to get from this. All right, so if we are going to go down the aerobic pathway and we have oxygen, we can now go into the second stage. So we went through glycolysis, we made 2 ATP. We're now going to go into another reaction called the Krebs cycle, right? And the Krebs cycle is kind of in a 2-step process as well, where we have a preparatory step and then we go through the cycle itself, right? So just like with glycolysis, we had to do some reactions first before it could get started. And that's kind of what we're doing here as well. So this is that pyruvic acid, or that pyruvate molecule, and it's going to combine with coenzyme A, and then that's going to alleviate CO2. That's where the CO2 comes from that we breathe out or bacteria give off when they're going through respiration, right? And again, NADH is going to convert to, or NAD plus is going to convert to NADH. And again, we utilize these molecules later. Just keep them in the back of your mind that these things are being made, all right? So we don't make ATP in this step, but we do make an electron carrier, and that's important. And also we convert this to acetyl-CoA, all right, which is this molecule here that's going to go into the Krebs cycle, all right? So now once we go through this reaction, we go into the big reaction, and that is the Krebs cycle. And this is the energy conserving step here because we're making ATP, all right? So we're going to go through, and basically sometimes this is called the citric acid cycle because citric acid or citrate is one of the products in here, and it's named that way in some texts, but usually it's called the Krebs cycle, all right? And it's named after the scientist that discovered it, Krebs. Now, notice what we're doing here as well. We're making some ATP. We make 2 more in this step, all right? So we convert ADP to ATP, but we're also creating more CO2. It's going to be a waste product that we give off. But more importantly, we're making these electron carriers, FADH2 and NADH. Just think of them as a molecule that's just got a little bundle of energy, an electron that it can carry somewhere else, all right? That's kind of the way to think about these. They're carrier molecules. They grab onto an electron and they give it to somebody else. So they're going to pass it down this electron transport chain later on, all right? And notice we produce a lot of these in this cycle. So we keep pumping these out. And again, this is going to go around twice, and we're going to produce ATP, 2 molecules of it, all right? So as we go through one time, we make one, and the second time we make another one, all right? But in total, we make 2 in this step. I'm going to go back and go back to way back here. Notice I said we made 2 in glycolysis. There they are. This is that Krebs cycle. So this is that preparatory stage. Then we go to the energy conserving stage and we make those 2 ATP right here. But notice all of these carrier molecules, they are going to be shuttled off into this electron transport chain. This is where most of our energy is going to be carried, right? And we're going to leave these other systems and come into here and then use that energy to make ATP, and we make a lot of it, right? We're going to make 34 molecules of it. So again, we make 2 up here, 2 in the next stage, and then we're eventually going to make 34 more in this electron transport chain. And that's what I'm about to get into. So let me move back over here. So again, that's where these molecules are going to go, into the electron transport chain. Right? So again, just think of them as a carrier of energy. They carry energy from one place to the other. It's like a gas can carrying a bunch of gas to put in another car. Right? Right, so again, we get a net of 2 ATP down here, but along the way, look at how many of these carrier molecules we're making. And these are going to add up, and we're going to do a little bit of math here in a minute, and I'll show you where those 34 ATP come from, right? So I told you these guys are important because they carry the energy that we're eventually going to use down here to make ATP, right? So again, we come in, we prep it by converting pyruvic acid to acetyl-CoA, and then that's going to go through the citric acid cycle or the Krebs cycle. And then while we're doing that, we produce 2 ATP along the way. All right. So again, we've already got 4 now that we have made. We made 2 in glycolysis. We have 2 more we've made in the Krebs cycle. Now we're about to go into the electron transport chain where we really make a bunch of ATP. All right. So this is going to occur in the plasma membrane of prokaryotes. And this is one reason prokaryotic organisms can make more ATP than eukaryotic organisms. Typically, we say eukaryotic make 36, whereas prokaryotic make 38. And it's because they don't have to waste ATP to shuttle molecules across the membrane of the mitochondria in the cell. They don't have a mitochondria. They are the energy maker itself. Remember, we think mitochondria used to be bacteria. This is one of the reasons we think that is the case, because bacteria can make their own energy. They don't have to have an organelle to do it for them. All right. So again, that's where we get the extra 2 ATP that we're talking about. Now, what's going to happen is these carrier molecules, these flavoproteins, cytochromes, and ubiquinones, these are going to be oxidized and reduced as electrons are passed down the chain. And you're going to notice that these lose energy as they go down. All right. And then we're going to make ATP by what's called chemiosmosis. And we're going to see how that works. We basically have an enzyme that almost looks like a mechanical wheel spinning around, and it's going to convert ADP into ATP. But we're going to have to set up a concentration gradient to do that. All right. And that's where these carrier molecules, that NADH and FADH2 and all of those come in. They're carrying the energy to fuel that enzyme. So these are the molecules that are going to come in and carry these electrons. Now notice my energy level goes down, and that's kind of what this is representing. Notice how energy here goes up. So we're really high energy state up here, but as we pass those electrons down, these cytochromes and things, we're going to lose energy. But by doing so, we're pumping hydrogen ions outside of the membrane, right? And then it's going to want to come back down through the membrane through an enzyme called ATPase, and that's where we start making all of this, ATP that we're going to produce. It's like a dam holding back water, and we allow the water to go through a turbine, and that generates energy. It's the same principle. We're allowing hydrogen ions to move through that turbine, that ATPase enzyme, and it's going to convert ADP into ATP, and it does so very efficiently, and it does it a lot. 34 ATP is going to be the output here. All right. So this is the electron transport chain. Notice that we have oxidation and reduction. Remember, that's just transferring an electron from one molecule to the other. So the one that releases the electron is oxidized, and then the one that receives the electron is going to be reduced. All right. So that's that redox reaction. Remember, you can't have one without the other. So one is oxidizing, the other is being reduced. But as we do that along this chain over and over again, we are pumping these hydrogen ions out. And as we do that, we create a concentration gradient where we have a lot of hydrogen up here and very little down here. And we all know back from anatomy class, hopefully you remember that things move from high concentration to low. So if we allow these to move through this ATPase enzyme, we're going to be able to use that energy to make ATP. Right? So chemiosmosis is where the electrons from NADH pass down the electron transport chain, and then we pump protons across the membrane. All right. So notice what we're doing here. We're pumping these hydrogen ions out, and then they're naturally, once they're in high concentration, going to want to move back down, and we allow them to move through this. Oops. Well, this is a better picture of it anyway. So we allow these hydrogen ions to move through this ATP synthase, which is an enzyme. And again, it actually rotates almost like a turbine. And as we do, we convert ADP into ATP. All right. So again, we're just going to keep pumping this. As long as we have hydrogen ions that are moving through, we can keep making ATP. And again, we'll just make it over and over and over again. All right. So this is what's called chemiosmosis. And this is allowing hydrogen to move through this enzyme to give it the energy it needs to create the reaction that forms ATP from ADP. All right. So again, as we pump these hydrogens through, that's where we make our ATP. Right? So we move from high concentration to low. So let's go back to this left side of this diagram. So you see all these NADH that is produced, and we're actually on this side. All of these that are produced in both of these cycles are going to go into the electron transport chain. Now we're going to do some math with this in a minute and show you how we get to that 34. I'm not going to make you calculate anything or whatever. I'm just going to explain to you where these come from. All right. So again, these high energy molecules that have electrons are going to go into this electron transport chain, and that energy is what we're going to use to make the ATP. So really the important thing to get out of this, glycolysis is making a little bit of ATP. The Krebs cycle is making a little bit of ATP. That's kind of a byproduct, right? The main goal of these cycles is to make these high energy carrier molecules that can go into the electron transport system, because that's where we can make a ton of ATP, right? So we're getting a little bit here, and we're getting a little bit here, but that's not the real goal. The goal is to create these molecules that can transfer that energy into this section so that we can make tremendous amounts of ATP. Right? So again, I've kind of robbed the picture here. This is actually a mitochondria in a eukaryotic cell, but pretend it's a bacteria cell because it's the same process. All right. So the final electron acceptor in the electron transport chain is oxygen, and oxygen is going to accept that electron, and that's going to be why we need oxygen to be in our system, because we have to have something to catch that electron at the end, and it's what's going to do that. All right. So again, the final electron acceptor is going to be molecular oxygen. All right. And again, we give off CO2 in the Krebs cycle. We give off water here. Those are what we breathe out as humans, and that's also what bacteria give off as waste products as well. Now remember, nature is in a really perfect balance. What really loves to use CO2 and water is plants, all right? And plants are going to be able to utilize our waste products. Plants give off a waste product called oxygen, and we utilize it, and so do bacteria. So we're in a balance with each other where they need our waste products and we need theirs. And we need them for food and everything else. So we're in a big cycle to where we kind of transfer all of these things back and forth, and we both benefit from it. All right. If plants were to go away tomorrow, we wouldn't have any oxygen, and all of these biochemical pathways would shut down and everything would die. Right. So it's a delicate balance we have between all of these organisms working together. And again, they've evolved together to do that. Oxygen is toxic in the atmosphere, so organisms develop ways of making it less toxic by including it in the metabolic pathway and then making it safe where it doesn't want to oxidize everything around it. All right. So again, this is kind of biochemistry 101. I won't get any further detail into the biochemistry, but just realize we're all dependent on each other as far as relying on the products that we make, even waste products, for us to survive. Right. So when we eat carbohydrates or bacteria consumes carbohydrates, the same process is going to happen. We're going to create the NADH and the FADH2, and these are the ones that are going to be able to create ATP for us. Remember, these are carrier molecules. They're carrying that energy. So for every molecule of NADH, we can make 3 ATP. For every FADH2, we can make 2 ATP. And I'll show you the math here in a second, but just kind of keep that in the back of the mind that NADH makes 3 ATP for each molecule and FADH2 makes 2, right? And that was where that math came up earlier, where he was doing the 1.5 and the 2.5. It came from these numbers, all right? So again, we're looking at it a little different mathematically, but it's the same equation, really. He just broke it down differently. All right, so here's where we do the math, and this is actually the better chart for you to look at to understand all of these processes, because it shows you the ATP production over here, but it also shows you the flow of electron, how many molecules of each thing we're making, and then also the hydrogen ions that are involved that we're going to pump across the membrane. All right, so let's go step by step through here, and you'll see where we get these 38 ATP. So glycolysis, again, we produce 2 ATP, 2 NADH, right? That then goes down into the Krebs cycle, and through the Krebs cycle on the way, we're going to produce 2 more NADH, and then the preparatory step, and then in the energy conserving step, we're going to make 6 more NADH and 2 FADH2. So if we look at our totals of all of these, we get 10 NADH, 2 FADH2. And if we do the math on that, you can see where we get the 34 ATP that are produced in the electron transport chain. Remember, for every NADH, we produce 3 ATP, so 10 of them will produce 30 ATP. 2 FADH will produce 2 ATP. Multiply that and you get 4. So we get a total of 34. Add back the 2 up here in the Krebs cycle and the 2 from glycolysis, and that gives us that 38 number that we see as a total. All right. So again, this is a really good diagram to show you a little bit of everything. In fact, I was going to study one diagram and have it for my test. I would have this one printed out. All right. So make sure you have this. You can use it on the test. I don't mind. I just want you to understand where these ATP are coming from. All right. These two and these two are just kind of being produced along the way, but we're really trying to make these guys. All right. Because when these guys enter this electron transport chain, we can produce a massive amount of ATP, and we maximize the amount of ATP we can get from 1 molecule of glucose. All right. This is in the handouts. Yes, I'm not sure if this exact one is. I think I put it in there. I'll double check and make sure, because this is the one that really sums everything up for you. All right. So you get your ATP total. Whoops. What we got? There we go. So you get your ATP totals, and you also get your totals of these, so that you know where all these ATP are coming from. And again, it's the energy that these are carrying that make all this ATP down here. All right. So notice we have oxygen accepting the final electron, and that ends the cycle. All right. We always have to have somebody to accept an electron. And we're going to see the same thing with fermentation. It's just a much simpler pathway, because we don't have to go through all of these steps, but we're not getting a lot of ATP out because of that, right? So we'll talk about that one in a few minutes. All right, so this is our cellular respiration formula. This is really the same formula for photosynthesis, is just backwards, all right? So let's take and go backwards with this, and let me explain what's going on. So during photosynthesis, we make ATP, all right? But this reaction is really what's going on inside of a plant. We're taking and using the energy from ATP and combining CO2 molecules or carbon atoms from CO2 to make glucose, all right? So if we reverse this reaction, that is essentially photosynthesis, all right? So photosynthesis runs this way, and we make a bunch of glucose, and then we eat something that has glucose and we run it the other way and go back and forth. So again, all these living things are tied together. Even this reaction is exactly the same, but in reverse in photosynthetic organisms versus heterotrophic organisms like us and bacteria, things that break down glucose, right? So if I'm a bacteria or a human or any other animal and I'm eating glucose, I am eating glucose that was originally made by photosynthesis, and a plant made that, right? And it took sunlight and used that for energy to create its ATP, and then it goes to what's called the Dark reaction to create glucose. And then we just simply reverse this reaction by taking glucose, combusting it with oxygen, and then we convert ADP into ATP, and we get our energy that way, right? So again, this is a reversible reaction. I wish they would have drawn it better, but this should be an arrow here going the other direction, showing you this can be reversed, because it is. Plants and other photosynthetic organisms are kind of the opposite of all of us heterotrophic organisms. Again, what they make, we use. What we make, they use, right? And we're in a delicate balance with them, all right? And again, all these reactions are very similar in all living things, right? Now, this is another good chart to look at. I'm pretty sure I included this as well, but this just tells you what's being produced in glycolysis in the Krebs cycle, and tells you how many ATP are going to be made from that, right? So again, we're going to be producing these NADH, FADH. That's where we're going to make the most of our ATP, right? Notice it tells you the electron transport chain is where these are going, even though it's occurring in different steps. They're all going to the same place. All right, so last thing we'll look at today is anaerobic respiration. And this is where we don't have oxygen available, all right? So the final electron acceptor now is not oxygen. It's going to be a high energy molecule like lactic acid or ethanol. Think of ethanol, for instance. We put that with gasoline because it burns just like gasoline, right? Not really burns hotter, but it's still got a lot of energy. That's what I'm getting at. So we're wasting a lot of the energy that's in glucose. All right. So by going through fermentation, all we are trying to do is replenish this NAD so we can get it back into the system to keep it going. That's all we're trying to achieve here, and get a little bit of ATP out, so we have a little bit of energy available. It's kind of a last ditch effort to get our energy. So again, less energy, we only get 2 ATP instead of 38. Right? So it stands a reason I would want to go aerobically if I can, right? But this is usually the last resort. So we can go through both lactic acid fermentation and alcoholic fermentation, right? Where we produce lactic acid or we produce ethanol. But the goal is the same, to replenish these NADH molecules and replenish that electron so that we can keep this system going, all right? And that's the whole goal of this, right? So fermentation releases energy from the oxidation of organic molecules. It doesn't require oxygen, and it doesn't use the Krebs cycle or the electron transport chain. And it uses an organic molecule as the final electron acceptor. And again, that's lactic acid or ethanol, right? And only small amounts of ATP are produced because the final product still has a lot of energy. Again, this is a can of ethanol. If you've ever had any kind of alcohol around, it's very flammable. The reason it's flammable is because it contains a lot of energy. We didn't pull the energy out of glucose to make ATP. We put it into this molecule, all right? And so this molecule still retains a lot of that energy. So let's say a glucose molecule, and I'm just throwing out even numbers here. Let's say it has 10 joules of energy, all right? This ethanol may have 8 joules of energy, and we put 2 joules of energy into the 2 ATP molecules, right? So we're not getting a lot out of glucose, and most of that energy is going to be wasted for this organism, and it's going to be producing a high energy molecule that it can't use, all right? But it's a last resort. We have no oxygen around. We need to get ATP somehow, and this is the only way we can do it. All right? So again, glycolysis is going to occur. We're going to make our 2 ATP. The pyruvic acid, or a derivative of that, is going to go into fermentation. again, we are going to produce NADPH plus. That line should be down here, but this NAD plus goes back up here and goes back into this reaction and starts the whole process over again with the next molecule of glucose. All right. So again, we're just trying to keep the system going, just pumping out a little bit of ATP, just enough to keep us alive. But we're going to go through this process over and over again to produce enough ATP to live. Right, so lactic acid fermentation produces lactic acid. We have homolactic fermentation where only lactic acid is produced. And we use these type of reactions or this type of fermentation to make food products because we don't want these other byproducts. We only want the lactic acid. Lactic acid is what makes yogurt, for example, tangy. It's what makes sauerkraut sour, right? It is that lactic acid that we're tasting in there, right? So as we take sugars and ferment them, in this case with lactobacillus, which is one that we use to make yogurt, that lactic acid then will go into the product and give it that tangy taste that we all know, right? So we make pickles this way, sauerkraut this way, kimchi this way, all those fermented products. So if you want to make one for extra credit, remember that's on Blackboard and you're utilizing this reaction. Now heterolactic, we produce lactic acid, but then we produce other compounds, right? So with this one, we may produce other compounds that we don't want, all right? That may be toxic, all right? You know, generally spoiled food's not good to eat, but if we have a homolactic fermenter, it's only producing the desired product and we're not making these other byproducts that make us sick, all right? And you're gonna see some of these other products we can utilize. We can use them industrially, but we don't wanna consume them. Now, alcohol fermentation produces ethanol and CO2. You know, I've made beer and wine and stuff for years, and you have to put what's called a fermentation lock on top that allows CO2 to bubble off, but not allow contaminants to get into your brew or whatever. So you add yeast, that yeast then will take sugars and convert that into alcohol. However much sugar you have in there is how much alcohol is going to come out as a result. But typically you can't make anything that's over about 14% alcohol. Alcohol is a toxin, and when the bacteria or yeast produce enough alcohol, it starts to poison them, and then they die off. So about 14% is kind of the limit that they can take, and then they start dying off and they stop fermenting at that point, right? The only way to get something above 14% is to distill it, to concentrate the alcohol by heating it up and cooling it back down and separating it from the water that may be in there. So again, fermentation, whether it's lactic acid or alcohol fermentation, we're just trying to produce these NAD plus to go back into the system up here so that we can keep this going and making ATP. So this is the stage that the bacteria wants to use to get ATP, but in order to rejuvenate this, we have to bring an electron back up into this stage so that we have the energy to actually do this, all right? And that's why they go through the production of lactic acid or ethanol, right? So again, glycolysis is where we make the ATP, and then the fermentation is strictly when no ATP is made here. We're strictly trying to get these electrons back up into the system. All right, so this is the same guy we watched a minute ago, and he's going to go over anaerobic respiration, the one he was referring to in the first video. Okay.
Hi everyone, Fritz here. In today's video, we're going to go over your anaerobic respiration. When we look at bacteria in a nutshell, the main objective of the bacteria is to produce energy. And whether this bacteria is an anaerobe or an aerobe, the primary process in which the initial ATP is made is always in an anaerobic fashion. And then after the initial ATP is produced, the bacteria, based off of its metabolic behaviors, will either continue down the anaerobic pathway or go down an aerobic pathway. And looking at those steps, the bacteria under normal circumstances will start off with a carbon source such as glucose, it will then put glucose through a process called glycolysis. At the end of the glycolysis process, we end up with 2 ATP molecules as well as 2 NADH molecules. Glycolysis will then convert the glucose to 2 pyruvate molecules, which are 3 carbon molecules each. And all of this is in a chemistry class. I just wanted to illustrate the fact that we are going to maintain the same amount of carbons going in as we do coming out. Now, if this bacteria is an aerobe, it will take the 2 pyruvate as well as the 2 NADH and put it through aerobic respiration, such as the electron transport chain, thereby creating anywhere from 32 to 36 ATP. However, we're focused on the anaerobic process. So what happens if the bacteria, which what we have here in this case, is an anaerobe? Well, in that case, the bacteria will go down the anaerobic pathway. And at the end of this process, the bacteria will either make alcohol or lactic acid. Either way, this process does not actually make energy, at least not enough energy worthwhile for us to count. But what's more important is that in this process of fermentation, the bacteria will actually take the 2 NADH from up here, convert it to NAD+, and then recycle it back into glycolysis. And the reason why this is so important is because these bacteria want to make the most use out of the resources that are available to it. So it doesn't want to waste anything, as was evident when we maintained our carbons from 6 up here to 6 down here. So this bacteria, if it wants to make 32 to 36 ATP as an aerobe will, it will actually have to repeat this process anywhere from 15 to 17 more times. And the most efficient way to do this is to recycle the NADH that it's not using and put it through glycolysis again, because it is a reactant in the glycolysis process when the bacteria finally makes the 2 ATP. But of course, for any one cycle of aerobic or anaerobic respiration, the aerobic will always create more ATP. But because this isn't available to the anaerobe, it has to go through this process. And here's some additional questions that you can have on test day.
All right. So again, he's going to go into trying to prep you for a test. That's kind of what he does on his videos. But that just kind of goes through and shows you, in a nutshell, both aerobic and anaerobic pathways. Right. So those are the 2 ways that a bacteria can make energy. That's it. That's the only 2 ways that they can gain energy from the environment. So we kind of generically use glucose as the sugar source, but it's really any carbohydrate. Right. So a lot of bacteria, if they have the right enzymes, can break down things like lactose and maltose and other sugars to get energy, but they carry it through the same process. Right. So again, this is the only 2 ways really that a bacteria can get energy, either not using oxygen and going through anaerobic fermentation, or having oxygen around and going through the aerobic. Sorry, it's all anaerobic up here, threw me out. So again, it prefers, if it can, to use oxygen and get that maximum amount of ATP out of that. All right, so these are some sources of different products that we use. And again, I mentioned earlier that a lot of times when we go through lactic acid fermentation, for instance, it can be heterolactic, where we can produce other things. All right, so we can take this pyruvic acid, and depending on which bacteria we use, we can produce different products. All right, so propionic acid, acetic acid, we can make butyric acid, butanol, acetone, isopropyl alcohol. All these things, we make on an industrial scale. We use bacteria to make them in a lot of cases. All right. Ethanol. Again, we utilize yeast to ferment corn, and most of our corn produced in this country actually goes into animal feed and making ethanol. We don't eat that much of it. All right. And the ethanol producing corn is not even edible. It tastes like crap. Nobody would eat it, but it has a lot more sugar in it so that we can ferment it and create ethanol from it. So again, when you go through the middle of the country, you see all these cornfields. Most of that's not ever going to be used by humans as food. We're going to utilize that to make ethanol through government subsidies and things. It's put in your gasoline. And again, we grow a lot of crops like that we don't actually necessarily eat. Right? We're using that for energy and other things. Right? So again, these are a few things, and again, you can see some of the microorganisms that are utilized to make those. Right? So again, it can be anything from a bacteria to a fungus. Remember, yeast are fungi. They're not bacteria. They're single cell, but they are fungi. Right? So again, this is that chart: 2 ATP, 2 ATP, 36 here. If we get through fermentation, we're only making the 2. Right? All right, I'm going to stop here, and I'm going to look at your quiz, and I think I have one ready to go. So if you do have to go to work or something, you should be able to finish. You've still got a few minutes. So let me cut off my recording and take a peek at the quiz, and I'll see